In the previous module, we walked through the basic steps of setting up a CFD simulation. Now let's see how such simulations can be applied to understand scaling issues, both in terms of their emergence and their impacts. Sometimes purely looking at the flow pattern can already be quite instructive. Poorly mixed zones may already be observed, and for cells sensitive to hydrodynamic stresses, such as animal cells, flow fields and the associated shear rates are very instructive. But for most microbial applications, we are interested in gradients of dissolved gas concentration and substrate concentration. Today we focus on the production of penicillin as an example, which is sensitive to gradients in glucose concentration. We will analyze this process in two distinct ways. First, from the reactor perspective, and later from the perspective of the microorganism. Although the process is aerated, for now we ignore the airflow to simplify the simulation. And here you see a simulation of a 54 cubic meter reactor with two impellers and the resulting flow field. In the flow field, you can already interestingly identify the formation of certain compartments. And mixing between these compartments may be poor and could be something to address. For validation, we have used the mixing time. A mixing time of 100 seconds was measured in practice for this reactor. The simulation gives us 80 seconds. Good enough considering the simplifications of ignoring airflow and also the fluid rheology. Now we add biokinetics to our simulation and add a glucose feed at the top. We consider the end of the fermentation with a high and fixed biomass concentration of 55 grams of dry weight per kilogram. Simple monokinetics are used to model glucose consumption. And knowing that the fermentation is glucose limited, we can compute a reaction time scale. This gives us 0.32 seconds, which means that we expect quite a gradient. And indeed, that's what we find. The concentration of glucose near the bottom is close to 0, while in the top, concentrations are in the millimolar range. The challenge now is for us how to interpret this information. We know that glucose consumption saturates for high glucose concentrations, and the region where this happens is indicated here in red. We also know that the organism Penicillium cryptogenum needs approximately 5% of the maximum glucose consumption for maintenance purposes. Below 5% uptake, it may get into a problem, starvation. That region we indicate here in blue. And the region in between, in yellow, is glucose limited, but there is sufficient glucose available. This excess regime in red covers about 7.5% of the volume, while starvation in blue covers over 50%. Now, if you were to scale this process down, you could attempt to mimic these fractions, either by coupling several lab-scale reactors to each other, each mimicking a zone, or by fluctuating the feed of a single lab bioreactor, such that cells are on average in excess conditions for 7.5% of the time and starvation conditions for 50% of the time. But what is the residence time of the cells in each of these zones? You can make an estimation based on the mixing time. The mixing time of 80 seconds means that cells cycle throughout the reactor approximately every 20 seconds. Or you can look at the flow rates between different zones, but this measurement can be very sensitive to the position. Also, these methods from the reactor perspective do not account for the distribution of residence times. So what we can do instead is take the cellular perspective, using a technique called Lagrangian particle tracking. By following the particles representing cells in the bioreactor in this simulated flow field, we can register what substrate concentration cells experience over time. We call this the lifelines. Such a lifeline provides information on how long cells reside in a certain reactor region. Here, the exposure to excess and to starvation is highlighted in red and blue respectively. In a CFD simulation, we may track thousands of particles in this way, and with many lifelines available, we can analyze how frequently cells transit between certain regimes, providing residence time distributions inside these respective regimes, which can be used to design a multi-reactor scale-down setup. But we can also conduct other types of analyses. If we zoom in on each of these lifelines, we can recognize long periods of starvation alternating with arc-shaped peaks. And in what we called arc analysis, we measure the duration and the magnitude of these peaks, as well as the intervals between them. This information can be used to design a scale-down system based on variations in the feed rate using our CFD analysis. We can provide information on the duration and the magnitudes that should be imposed. Well, so far, we consider how heterogeneity manifests, but how does it impact cells? And to answer that, we need to include a reaction model that captures the intracellular response, particularly the penicillin production in this case, because we're interested in this particular phenomenon, and we need to capture how it responds to extracellular variations in conditions. This model needs to account for the intracellular state of the organism, because we cannot assume cells instantaneously respond to their environment. For penicillin, we use a model with 9 intracellular pools, of which 5 metabolite pools and 4 enzyme pools. Most relevant for us are the glycolytic intermediates, and of course the penicillin-producing enzyme pool. The glycolytic intermediates are the readily available sugars, and these eventually regulate the penicillin enzyme production rate. Too little, and the energy-intensive enzyme production slows down. Too much, and production is also repressed. The timescale of variations in the glycolytic intermediates is approximately 10 seconds. So they buffer the rapid variations in extracellular glucose, but the intercellular glycolytic intermediates still go up and down quite fast as the cell moves throughout the reactor. Conversely, the enzyme concentration responds on the timescale of hours, so we expect the enzyme level to end up constant, insensitive to rapid extracellular variations. The question is, at what level will the penicillin production capacity level off, under the pressure of continuous regulation? To use these models, we need the Lagrangian particle-based approach because we need to keep track of the history of many cells to see how they're adapting to the constantly changing surroundings. So the reaction model is linked to these individual particles in the simulation. Now let's have a look at how these organisms behave. In this video, organisms are colored by the level of glycolytic intermediates. Red means that the glycolytic intermediate pool is full, and blue means that it's nearly empty. You can see the levels go up and down, and clearly, typically the red particles are found near the feed in the top, while the blue particles are found near the bottom. But, as expected, the cells are never quite in equilibrium with their surroundings. Under these variable conditions, our simulation predicts that the penicillin production rate and yield is 33% lower than compared to the ideal homogeneous lab conditions. Note, by the way, that this would be 85% if we did assume instant equilibrium between the cells and the extracellular glucose. So clearly we cannot ignore the timescales of metabolic adaptation. Now, the combination of CFD and metabolic modeling can be a powerful tool to analyze the impact of scaling effects. In this case, we note quite a strong heterogeneous environment, and we can use our simulation to predict how strongly cells respond to that. Whether this 33% decrease in yield that we observed is acceptable, of course, depends on the economics of your process. But at least now you can take it into account. But we can also go a step further and use our simulations to analyze the effect of process changes. What if we move the feed from the top to a more favorable location with a high velocity close to the center, the outflow of the middle impeller? We see that the axis region now is nearly gone and the starvation zone is split in two, and the residence time in both of these extreme zones are decreased. As a result, the intercellular variations that we observe are reduced, and the predicted yield loss is now only 9%. Now we must keep in mind that these predictions are purely computational, although the kinetic model and the CFD models have in themselves been validated with experimental data from research experiments in their own right. Detailed data from the industrial process is unfortunately still lacking. and several physical simplifications have been made in the setup of our simulations. Nevertheless, comparing our model predictions with fed batch production data did show a favorable prediction if we compare it to more traditional black box ideal mix models. And as such, we expect CFD methods to become more popular, and we expect further comparisons with real production data for various systems, as well as real downscaling results to further show the capabilities and limitations of these methods in the context of scale up and scale down. The approaches that we outlined here are very novel, and they're not yet applied very widely. Several researchers, including ourselves, are working on making them more fast, accessible, and more accurate. But the potential is clear. In case there are scaling effects, CFD allows us to quantify their impact, evaluate mitigating actions, and hopefully decide the optimum between economy of scale and negative scaling effects.